An incredible diversity of life exists on Earth. Jellyfish that drift in the sea like ancient ghosts, the electric-colored fish that inhabit coral reefs, the screaming parrots and sluggish koalas that inhabit Australian forests, and the bison that once dominated the Great Plains of the United States are just an infinitesimal sampling of the Earth's living diversity. In spite of their diversity of forms, all the living creatures of the Earth share a common molecular ancestry. All contain long molecules of DNA and RNA that carry encoded with them an individual's unique blueprint. All metabolize glucose and other sugar molecules for energy. All the cellular membranes of all the cells of all the creatures on Earth are made of long-tailed phospholipids. And all the cells in the living world contain large molecules called enzymes that build other molecules. And all these molecules have molecular skeletons made of carbon atoms. Molecules like DNA, glucose, and phospholipids are called organic molecules because it was once thought that only living organisms could create molecules with carbon skeletons. The Earth's living organisms are made up of an amazing variety of organic molecules. For example, the horns, coat, and hooves of buffalo are composed of organic molecules, as is the nectar of flowers and the trunks of trees. In fact, nearly all the substances found in living organisms, other than water and ions, are organic. It is the ability of organic molecules to manufacture other organic molecules, to be modified into new forms and take on new functions, that has allowed life on Earth to develop and evolve. The form and functioning of our bodies is dependent on an incredible array of organic molecules: proteins, amino acids, nucleic acids, triglycerides, carbohydrates, and a variety of other complex molecules. Though it was originally thought that only living organisms could create organic molecules, today, organic chemists routinely synthesize organic molecules in the laboratory. As a result, organic molecules are now simply defined as molecules that contain carbon atoms. And it is to carbon atoms that organic molecules owe their versatility. Carbon atoms have 4 electrons in an outermost energy level that becomes stable with 8. Carbon atoms thus become stable by forming single, double, and triple covalent bonds with other atoms, until 4 pairs of electrons are shared between the carbon and other atoms. Molecules made up of a skeleton of carbon atoms can assume a variety of complex shapes, including rings, branches, chains, and a nearly infinite number of 3-dimensional shapes. While carbon atoms form the skeleton of most organic molecules, other groups of atoms, called functional groups, are attached to the carbon skeleton. It is these functional groups that largely determine the characteristics and chemical activity of various organic molecules. Many organic molecules are made up of hundreds, if not thousands, of atoms. Rather than being synthesized atom by atom, these large organic molecules are assembled from smaller molecules called monomers. Molecules built from monomers are called polymers. For example, amino acid molecules built out of 4 different functional groups attached to a single carbon atom are monomers. The joining together of amino acids forms proteins, which are polymers. Almost all biological molecules are formed by chemical reactions called dehydration reactions. As monomers are joined in a dehydration reaction, a hydrogen ion is removed from one monomer and a hydroxyl ion from the other. The hydrogen and hydroxyl ions combine to form a water molecule. Thus, dehydration reactions derive their name from the fact that the reacting molecules give up a water molecule and are therefore dehydrated. During hydrolysis reactions, which break apart organic polymers, water molecules are also broken apart. One of the new molecules, created by a hydrolysis reaction, receives a hydrogen ion from a split water molecule, while the other receives a hydroxyl ion. Nearly all the thousands of different monomers and polymers fall into one of four categories: carbohydrates, lipids, proteins, or nucleic acids. Let's now look at the members of these four categories and at the roles they play in living organisms. All carbohydrates are built from molecular monomers, or subunits called sugars, that contain only carbon, hydrogen, and oxygen atoms. The typical sugar has a backbone of 3 to 7 carbon atoms. Most of these carbon atoms have a hydrogen group and a hydroxyl group attached to them. Thus, the general formula for sugars is CH2ON, where N is the number of carbons in the backbone. It is from this formula that carbohydrates derive their name, which literally means carbon and water. When dissolved in a watery environment such as the cytoplasm of a cell, the carbon backbone of a sugar usually circles up into a ring. It is in this ring form that sugars are linked together by dehydration reactions to make larger carbohydrate molecules. Carbohydrates fall into one of three categories, depending on the number of sugars they contain. Carbohydrates, such as glucose or fructose, that consist of a single sugar monomer, are called monosaccharides. Sucrose, lactose, and other carbohydrates that contain two sugar monomers are called disaccharides. Starch, glycogen, cellulose, and other carbohydrates made out of long chains of sugars are called polysaccharides. Glucose is the most common monosaccharide and one of the most abundant sources of chemical energy in the living world. Glucose is the sugar found in the highest concentration in the blood of animals, and our own central nervous systems are critically dependent on glucose for energy. But glucose molecules are also the building blocks for the polysaccharide cellulose. Cellulose is the major structural material in the trunks and branches of trees. Glucose's chemical formula of C6H12O6, or CH2O6, is shared with fructose and galactose. However, the three have different chemical structures, giving each a different taste. Monosaccharides, such as glucose and its relatives, have a short lifespan in the cell. Most are broken down to provide energy or linked together by dehydration reactions to form disaccharides or polysaccharides. Two other important monosaccharides are ribose and deoxyribose, each of which have 5 carbons. The two molecules are identical, except that, as the prefix deoxy implies, deoxyribose molecules have 1 oxygen less than ribose molecules. Ribose and deoxyribose form critical parts of RNA and DNA molecules respectively. RNA and DNA are critical in passing the blueprints of life from cell to cell and from generation to generation. Disaccharides are formed by linking two monosaccharides. Cells, especially those of plants, use disaccharides to store and/or transport energy. When an organism needs energy, disaccharides are broken apart into their monosaccharide subunits by hydrolysis. Sucrose and lactose are two common disaccharides. Each molecule of sucrose, or table sugar, has 1 glucose and 1 fructose molecule. Sugarcane and sugar beets produce and store huge amounts of sucrose and are harvested commercially to produce table sugar. Many female mammals produce lactose, or milk sugar, by adding a galactose molecule to a glucose molecule to provide energy for their nursing offspring. In order to store energy, living organisms join monosaccharides, usually glucose, to form polysaccharides, such as starch in plants or glycogen in animals. About 20% of starch molecules, which are commonly found in the roots, seeds, and tubers of plants, occur as coiled and branched chains. These chains can be up to 1,000 glucose molecules long. The remaining 80% or so of starch molecules consist of branched chains, each branch made of up to 500,000 glucose molecules each. Potatoes are the quintessential example of starch storage in plants. The tubers of potato plants, which we call potatoes, are made up nearly exclusively of cells storing large amounts of starch. Glycogen provides long-term energy storage in animals. Mammals and other vertebrates store glycogen mainly in their livers and muscles. Glycogen molecules are usually much smaller than starch molecules, having branches every 10 to 12 glucose molecules. The frequently branching structure of glycogen makes it easy for glucose molecules to be split off the glycogen polymer rapidly. This provides the quick access to energy required by many animals. Polysaccharides are also used structurally. Probably the most familiar structural polysaccharide and the most abundant organic molecule on the face of the earth is cellulose. Cellulose makes up a large part of the cell walls of plants and about half the bulk of tree trunks. Biologists have estimated that the fields and forests that cover our earth synthesize over 1 trillion tons of cellulose each year. Humans harvest a significant percentage of this cellulose to provide building materials, paper, and other products. Cellulose, like starch, consists of glucose subunits bonded together. However, in cellulose molecules, the bonds between glucose monomers continually reverse so that every other glucose molecule is upside down compared to the next. This difference in orientation prevents the digestive enzymes of most animals from attacking the bonds between the glucose monomers and using the tremendous amounts of energy available in cellulose. However, there are a few microbes, such as those found in the digestive tract of termites, cows, and certain other herbivores, that have enzymes that can digest cellulose and break down these bonds. As a result, cellulose is food for these microbes. The termites, cows, and other herbivores that host these cellulose-digesting bacteria appropriate some of the high-energy molecules produced by the microbe's digestive process for their own nutritional use. Polysaccharides are also used as a starting point for the synthesis of other structural materials, such as chitin. Chitin forms the hard outer covering or exoskeletons of insects, crabs, and spiders. The cell walls of many fungi also contain chitin to provide stiffness. Chitin has the same alternating upside-down bonding scheme as cellulose. However, the glucose monomers are modified in chitin. A unique nitrogen-containing functional group replaces a hydroxyl group in each glucose monomer in a chitin polymer. Modified polysaccharides are also found in the cell walls of bacteria, the lubricating fluids in our joints, and the transparent lenses of our eyes. There are many other molecules within plants and animals made up partially of carbohydrates. For example, as we saw earlier, RNA and DNA molecules use the carbohydrates, ribose and deoxyribose, to help form their intricate structure. Other carbohydrates attach to proteins to form glycoproteins that serve as identifying markers for different cells, like our own red blood cells. The glycoproteins that are or are not found on the cellular membrane of our blood cells determines our blood type. A diverse assortment of molecules are classified as lipids. Fats, which store energy and provide insulation, are lipids. Phospholipids are the major component in cellular membranes. Waxes, which provide a waterproof coating for a variety of animals and plants, are lipids. Certain hormones, called steroids, are also lipids. Steroid hormones control various life processes, such as the development of secondary sexual characteristics in many vertebrates and molting in insects. In spite of their diversity, all lipids share a basic feature: long sections composed almost entirely of non-polar carbon-carbon or hydrogen-carbon bonds. This non-polarity makes lipids insoluble in water. Lipids are divided into three groups. One group contains oils, fats, and waxes, which are similar in structure and contain only carbon, hydrogen, and oxygen atoms. A second group, phospholipids, are structurally akin to oils but contain phosphorus and nitrogen atoms in addition to carbon, hydrogen, and oxygen atoms. The third group consists of steroids. Steroids are made up of 4 fused carbon rings to which various functional groups are attached. Let's look at each of these groups in turn. Fats and oils are formed by dehydration reactions between a glycerol molecule and 3 fatty acid molecules. Glycerol molecules are short 3-carbon molecules with 1 hydroxyl group per carbon. Fatty acids consist of long carbon and hydrogen chains with a carboxyl group at the end. It is the combination of 3 fatty acids and 1 glycerol molecule that gives fats and oils their chemical name, triglycerols. Fats and oils contain a high concentration of energy, 9.3 kilocalories per gram. This is over twice the 4.1 kilocalories of energy found in a gram of sugar or protein. Bears and other animals use fat molecules as a semi-permanent energy storage. During the late summer and fall, bears and other large mammals often eat non-stop to put on fat to tide them over during winter hibernation or migration. At room temperature, fats are solid, while oils are liquid. This is explained by a difference in the fatty acids that make up fats and oils. The fatty acids and fats are joined exclusively by single bonds between the carbon atoms making up their chains. Hydrogen atoms attach to the remaining 2 bonding sites on each carbon atom. Carbon atoms joined by single bonds form straight lines and allow the maximum number of hydrogen atoms to bond to them. As the maximum number possible of hydrogen atoms are bonded to these fatty acids, they are referred to as saturated fats. The straight chains of saturated fatty acids found in fats allows them to easily nestle together and form a solid at room temperature. Unsaturated fats result when 1 or more double bonds occur between carbon atoms in a fatty acid, causing fewer hydrogen bonding sites to be available. Unsaturated fatty acids are usually found in oils, such as olive and corn oil. The double bonds between carbon atoms in the fatty acid chains of unsaturated fats cause the chain to kink at a sharp angle. The kinks caused by the double bonds prevent unsaturated fats from clumping together and forming a solid at room temperature. Waxes, unlike oils, contain only 1 fatty acid chain. Attached next to this fatty acid chain is a long-chain alcohol. This combination makes waxes very resistant to penetration by water. Thus, waxes often provide a waterproof coating for the leaves and stems of land plants that prevent water from escaping through evaporation. Many animals also synthesize wax. For example, some mammals produce wax to waterproof their fur, and many insects waterproof their exoskeletons and wings with a coating of wax. Bees synthesize wax and use it to create elaborate honeycomb structures in which they live and store honey. Phospholipids are a second group of lipids. Phospholipids are a major component in the building of cellular membranes. Phospholipids are like oils, except that a phosphate group replaces one of the fatty acids and faces in the opposite direction of the other two fatty acid tails. The end, or head of each of these phosphate groups, is polar. Since they are polar, the heads of phospholipids are soluble in water, unlike the insoluble fatty acid tails. During the formation of cellular membranes, phospholipids arrange themselves into two layers, called a phospholipid bilayer. The polar, water-soluble or hydrophilic heads face the watery cytoplasm or extracellular fluid, while the water-insoluble or hydrophobic tails hide inside the bilayer. The structure of steroids is substantially different from that of either phospholipids or oils, fats, and waxes. Steroids, as we noted earlier, are composed of 4 molecular rings, to which various functional groups are attached. The sex hormones, testosterone and estrogen, are both steroids. Variations in testosterone and estrogen levels are responsible for many of the anatomical and physiological differences between male and female vertebrates. The use of another group of steroids, anabolic steroids, among athletes in sports such as football, bodybuilding, and weightlifting has been the subject of much controversy. Anabolic steroids promote the storage of protein and the growth of muscle and other tissue. However, they are often associated with devastating side effects, such as liver damage, cardiac problems, aggressive behavior, and infertility when abused. The steroid cholesterol is a vital component, along with phospholipids, in cellular membranes. But too high a cholesterol level in the bloodstream is a health threat. Cholesterol and other lipids can slowly build up plaque along artery walls, causing a condition called atherosclerosis, which often results in a reduced flow of blood to the heart, brain, and other vital organs. Pieces of plaque can also break off suddenly and totally cut off the flow of blood to a heart muscle, resulting in a heart attack, or to part of the brain, causing a stroke. Proteins are formed by dehydration reactions that string several to thousands of amino acids together in a chain. Proteins perform many functions in living organisms. Some proteins, called structural proteins, are used by organisms for structural purposes. The horns, hooves, and furs of mammals, for example, are made up of structural proteins, as is the silk used by spiders to spin their webs. Proteins also carry out a variety of other functions. The protein hemoglobin, found in red blood cells, transports oxygen to and carbon dioxide away from body cells. Special proteins called channel proteins control the flow of certain molecules across cellular membranes. The contraction of muscles is the result of the interaction of two proteins, myosin and actin. And the venom of snakes, scorpions, and certain spiders contains lethal proteins. However, the majority of proteins are enzymes. Enzymes control and catalyze most chemical reactions occurring in living organisms, often causing them to occur billions of times faster than they would normally occur. The dehydration and hydrolysis reactions that create and break down other molecules are carried out by enzymes. Thousands of different kinds of chemical reactions occur within cells, and any given type of enzyme is involved in only one or two of these reactions. As a result, even simple cells often contain 2,000 different kinds of enzymes. As we will see shortly, the shape of enzymes and other proteins is very critical to their proper functioning and the speeding up of reactions. All told, there are approximately 30,000 different proteins found in the human body. These 30,000 different proteins are assembled from only 20 different amino acids. All amino acids have the same basic chemical structure. A central carbon bonded to 4 different functional groups: a nitrogen-containing amino acid group, NH2, a carboxyl group, a hydrogen group, and a variable group represented by the letter R in structural equations. It is the variable group that gives each amino acid its distinctive properties. The dehydration reactions that build proteins out of amino acids are carried out in cellular structures called ribosomes. Enzymes in ribosomes join the nitrogen of one amino acid group to the carbon atom on the carboxyl acid group of another amino acid. As in all dehydration reactions, one H2O molecule is formed for each amino acid added to the chain. Ribosomes know in which order to place the amino acids in a protein from assembly instructions copied from DNA molecules and transmitted to them by messenger RNA molecules. The bonds between amino acids are called peptide bonds. Chains of 2 amino acids are called dipeptides. 3 tripeptides and 4 or more amino acids linked together are called polypeptides. The proteins found in living organisms vary in length, from 3 amino acids to chains thousands of amino acids long. The typical protein is a polypeptide made up of about 200 amino acid molecules. The shape of proteins can vary tremendously, depending on the order of the amino acids and the environment surrounding the protein. The peptide chains in the silk protein fibrin run side by side, creating molecules that look like long, pleated sheets. The amino acid groups in the protein keratin form a coiled shape called a helix, like that found in DNA and RNA molecules. Bonds called disulfide bridges between functional groups containing sulfur atoms often create sharp turns or irregular shapes in a helix. The complex shape of the proteins hemoglobin and insulin are created in part by disulfide bridges. The function of an enzyme is related intimately to its shape and structure. Nearly all enzymes are proteins with complex 3-dimensional shapes. Every enzyme has a dimple or groove, called the active site, to which reactant molecules, called substrates, attach. The active site of an enzyme is usually very specific, allowing only a few types of molecules or substrates to attach. The size, shape, and electrical charge of an enzyme's active site forces a substrate or substrates to enter the site in a specific orientation. After substrates enter the active site, both the substrates and the active site change shapes as a result of chemical reactions. The amino acids that form an active site often form temporary chemical bonds with the atoms of a substrate molecule. These bonds are used to break a substrate molecule apart or join it to another substrate molecule. After the reactions between substrates and enzymes are completed, the altered substrates no longer fit properly in the enzyme's active site and are expelled. The enzyme's active site reverts to its original shape and charge, ready to receive new substrates. Many poisons, including some insecticides, enter the active site of enzymes essential to brain function. However, as they don't react with other substrates, the poison molecules remain in the active site. As a result, critical reactions the enzymes promote can't take place. Nucleic acids are a 4th major type of organic molecule. As we alluded to earlier, genetic material found in the nuclei of each cell specifies the sequence of amino acids in every protein manufactured by that cell. The genetic material of living organisms is made up of nucleic acids. Nucleic acids are assembled out of monomers called nucleotides. Nucleotides have 3 parts: a phosphate group, a 5-carbon sugar, and a nitrogen-containing base. There are 2 distinct types of nucleotides: deoxyribose nucleotides and ribose nucleotides. Deoxyribose nucleotides contain the 5-carbon sugar deoxyribose and one of 4 different nitrogen bases: adenine, guanine, cytosine, or thymine. These nucleotides are used to form the nucleic acid DNA, or deoxyribonucleic acid. Ribose nucleotides contain the 5-carbon sugar ribose and one of 4 nitrogen bases: adenine, guanine, and cytosine, just as they are found in deoxyribonucleic acids. However, in RNA, the nitrogen base uracil replaces thymine. In DNA and RNA molecules, nucleotides are strung together in long chains formed by covalent bonds between the sugar of one nucleotide and the phosphate of the next. RNA usually occurs as a single strand, formed in this manner. DNA, on the other hand, is usually found in the nucleus of cells with 2 strands, bound together by hydrogen bonds between the nitrogen bases. However, only adenine and thymine bases and guanine and cytosine bases can bond together. Because of the different chemical bonds and monomers that make up DNA molecules, they take on the appearance of a twisted ladder, or what scientists call a double helix. DNA molecules can be millions of nucleotides long. Each long, complex DNA molecule in the nucleus of a eukaryotic cell is called a chromosome. Chromosomes contain subdivisions called genes. The sequence of nucleotides in most genes contains a kind of biological Morse code that has the instructions for building one complete protein. As we saw earlier, these instructions are copied onto RNA molecules that travel into the cell's cytoplasm and there direct the synthesis of proteins by ribosomes. There are nucleotides that are not part of DNA or RNA molecules that also play an important role in the functioning of cells. Cyclic nucleotides, such as cyclic adenosine monophosphate, or CAMP for short, are used as intracellular message carriers. If, for example, a hormone in the bloodstream, like adrenaline, attaches to a receptor protein on the cellular membrane of the cell, it may direct the synthesis of a CAMP molecule. The CAMP, in turn, directs an enzyme to synthesize more glucose in anticipation of the cell's increased energy requirements. A nucleotide frequently mentioned in the study of biology is adenosine triphosphate, or ATP for short. ATP, as its name implies, has 3 phosphates instead of just 1, like most other nucleotides. ATP molecules are unstable molecules that collect and store the energy produced by processes such as cellular respiration and photosynthesis. They then transport the energy to different structures in the cell to power their operation. For example, the energy stored in ATP molecules powers protein synthesis by ribosomes and the interaction between actin and myosin molecules that enable a weightlifter's muscles to expand and contract. As we've seen, our bodies and those of all living organisms contain thousands of different molecules, which carry out a wide variety of functions. Fat and sugar molecules, for example, store energy, while DNA and RNA molecules store information. Some large complex protein molecules build other molecules and speed up chemical reactions, while other proteins are used to build essential body structures or to control the flow of materials in and out of cells. No matter how large or unique, all these large molecules, or polymers, are built out of small molecular building blocks called monomers and fall into one of 4 categories: carbohydrates, lipids, proteins, or nucleic acids. While all living organisms share the majority of these different molecules, The exact combinations and concentrations vary between species and individuals. Thus, all the life that surrounds us is formed from shared molecular building blocks. It is simply variations in the molecular assembly instructions contained in DNA that gives rise to the incredible diversity we see expressed in the living world. The molecular instructions that have given rise to we humans have provided us with the gifts of consciousness and forethought, gifts that can be used to preserve the diversity found in the living world so that it can be enjoyed and studied by future generations.